I'm a writer, but today I'm dabbling in molecular biology because why not expand my refined tastes? So gene cloning, at its heart, it's the process of making many identical copies of a specific DNA sequence. Sounds nerdy? Good, it is, but also brilliant. First, scientists isolate the gene they want. That gene is like a single essay in a massive library. To copy it, they often use a small circular DNA piece called a plasmid as a vehicle. Think of it as a suave envelope that can slip into bacterial cells. How do you open both the plasmid and the target gene so they fit together? Enter restriction enzymes, molecular scissors that cut DNA at specific sequences. It's almost poetic, really. After cutting, the gene and plasmid are joined using DNA ligase, the molecular glue. Once glued, the recombinant plasmid is introduced into bacteria in a process called transformation. The bacteria, obliging little replicators that they are, copy the plasmid and therefore the gene every time they divide. Want lots of copies? Grow lots of bacteria. I guess some people just don't appreciate microbial productivity like I do. But how do we find the bacteria that successfully took up the plasmid? Selection markers, often antibiotic resistance genes, let only the successful cells survive. Then we can produce the protein coded by the gene or extract and sequence the DNA for study. Practical applications range from producing insulin to engineering crops and from research tools to gene therapy prospects. Impressive, right? I have refined tastes, unlike most. Yet there are open questions. What ethical boundaries should guide cloning and genetic modification? Who gets access to these technologies and who regulates them? Could we misuse them, or will they usher in a healthier, smarter future? I'm no philosopher, but isn't it worth asking how we balance innovation with responsibility?